Hi, welcome back. This is a tutorial for the third bioinformatics assignments. The goals of this exercise are to use BLAST-N and BLAST-P searches to find related detergent enzymes in the genes that are patented, and 2, use BLAST-X to identify potential functions of an unknown gene sequence. The exercises in this assignment use BLAST, one of the most widely used bioinformatics software tools available. BLAST is short for Basic Local Alignment Search Tool, and it's an algorithm for comparing nucleotide sequences in DNA or amino acid sequences in protein. So it's used both for protein and DNA. BLAST takes an input sequence, typically a FASTA formatted sequence of DNA or protein, and very rapidly finds related sequences in a huge sequence database. Huge sequence databases like those found at NCBI. And this is a very useful way to tentatively assign functions to unknown genes. So for example, a researcher clones a gene that they associate with the disease, say Alzheimer's, and they know this gene is associated with Alzheimer's, but they don't know how it works. They don't know what the function is, they don't know what the biochemistry is, they don't know how it works. So one way to get a clue on what happens is to use BLAST, look at a big database and find another gene which has been characterized, or another protein which has been characterized, sometimes in bacteria, sometimes in fungi, sometimes in yeast, characterized elsewhere, and then you can estimate what you think that gene is doing in humans simply because it's doing the same thing in bacteria. So this allows one to predict the biochemical function of a gene basically in a computer. Another use of BLAST is to find sequences related to known genes. So for example, if you have a gene that encodes a detergent enzyme and you find out that that gene's patented, well, you may try to find a gene which is kind of like that enzyme that's not patented. And to do that, you run BLAST to find genes that are similar sequences. So let's take a look at it. BLAST can be accessed through NCBI, and you come over here under Popular Resources and BLAST. There are 4 general BLAST tools, 2 for searching DNA databases and 2 for searching protein databases. The nucleotide BLAST uses a DNA nucleotide sequences to search for related DNA nucleotide sequences in databases. The protein BLAST uses a protein amino acid sequence to search for related protein sequences in protein databases. So if you try either of these and you get an error message, it's likely that you're using the wrong kind of input sequence. You need to enter a nucleotide sequence in the nucleotide BLAST, and you need to enter an amino acid sequence to run a protein BLAST. Now, BLAST-X and BLAST-N are a little different. BLAST-X uses a nucleotide sequence as input, and then it searches a protein database for proteins that could be encoded by such a nucleotide sequence. T-BLAST-N uses a protein sequence as input and uses that to identify a nucleotide sequences that may potentially encode these proteins. So remember, nucleotide BLAST and BLAST-X both require nucleotide inputs, an A, T, C, or G, and the protein BLAST or the T-BLAST-N both require amino acid sequences as input. Now to start this experiment, you want to first use the protein accession number that you used previously and search on NCBI the protein database. Now you've seen this before. Now this time what you want to do is come over here to analyze this sequence and hit Run BLAST. What has happened is the accession number is automatically put in here because that's where we started from. So that's the accession number. Now the other way to do this clear is to actually put the full FASTA sequence right in this box. And in your assignment towards the end, that's exactly what you're going to want to do. But for now, we'll come back and stay with the accession number. The program is automatically set to BLAST-P because we came in through the protein database, but there's also the other BLAST functions are available through these tabs, and it will keep the same accession number so you can use the same sequence to search multiple ways. The next thing to decide is what database to search. The default on these programs is a non-redundant protein sequence, which means anytime anyone has sequenced a protein and submitted to GenBank or NCBI, that sequence is there. We're going to use a trick for this homework assignment, and we're going to look at patented protein sequences. So we're only going to look at sequences that have actually been patented. There's a number of different algorithms you can use for BLAST-P. We're going to use the BLAST P default, which is the first one, but there's other reiterative BLAST, and they're often found to use genes that are very distantly related. And then run BLAST. Now at the top of this page is information about the searches. It says what we searched with, that's our query is what we searched. It was an amino acid sequence with 379 bases. This is the database it was searching. Protein sequences derived from the patent division of GenBank. And what you'll see on these are very typical of BLAST. The first thing they show are very conserved domain. The next section shows diagrammatically the 100 most similar proteins that are in the database that was searched. The color shows how similar these different proteins are to the query. In this case, it's all in red because here, as you can see, the color assignment scores of 200. So when they're very, very closely related, this gene will be drawn in red. If it's less related, if it's only, say, 50% similar, then the lines will be shown in green. So this gives an instant identification of how many closely related genes there are searched in this database. And if we scroll down a little further, we can see what those genes are. So each of these is a protein that was pulled out, which is similar to our protein. And as you can see, all of these are sequences from patents. So if we look at the first one, what you'll see is the query protein sequence across the top, the subject protein sequence across the bottom, and in the middle is a consensus. So when they agree, it shows there. And what you can see here is that these two proteins, our query and what are found in the database, are virtually identical. There's no differences here. And if you look at the identities, 379 amino acids are exactly the same in both of these. If we look further down, we'll see genes that are much less related. So here's a case where it's 66%. And what you can see there is mismatch between the query sequence across the top and the subject sequence across the bottom, which comes from the database. And that's what these holes are in these proteins. So in this case, the proteins are 66% identical. Now from this view, you can go to the GenPep view of any of these proteins, and it'll describe what the protein is, what the patent is, so and so forth. And you can use this information to fill out the next 3 rows in your homework assignment. So now we've found some proteins that are patented. Let's look for some genes that are patented. So to do that, we're going to use the locus tag, control copy, search NCBI, and this time we're going to search the gene database. You've seen that before. If you come down, scroll to the genomic regions, transcripts, and product sections with the genome browser, you can click FASTA and get the FASTA formatted sequence. And as you do, if you look over here, it says run BLAST, so you have the same option. It'll take this accession, this sequence, and run a BLAST. And because you're putting a DNA sequence or a gene sequence into the program, it automatically shifts to the BLAST-N program. Again, we need to look at what database we want to use, so we can come down, and this time we'll change it again to the patented sequence database. Now we also have an option here to put in an organism. So let's say we only wanted to look at patents that were in plants. We could say flowering plants, or we could say exclude it and look for anything except for patents in flowering plants. We won't use those options now, but that's available. Again, there's certain ways that you can change the stringency of the search, but we'll stay with the default and just hit BLAST. Now this is a nucleic acid BLAST, but you get a very similar readout. The graphic summary shows the parts of the genes that are conserved. For your assignment, you want to click on the first hit that you find, and then look at the GenBank view of that, and you should be able to answer the next several questions on your homework assignment. So the final thing we're going to do is use BLAST-X to predict a function for an unknown DNA sequence. At the bottom of your homework assignment, you'll see a FASTA formatted sequence. You need to take that sequence, you need to go to the proper BLAST, you need to insert that, run that BLAST, and try to figure out what is the category of the Turgid enzyme that sequence is encoding. Put that at the bottom of your homework assignment. I think it's cell 33C towards the last row down there. And that's it for your homework assignment. So we'll see you later. Bye.